Hello, everyone, and welcome to today's webinar titled N-GLYcanyzer, an automated process analytical platform to enable continuous manufacturing of biologics. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Antonina Salcido of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of the navigation bar or from the lobby of the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speakers. Dr. Áron Györgypál, Research Associate, Bioprocessing and Glycomics Chemical and Biochemical Engineering at Rutgers, the State University of New Jersey, and Dr. Shishir Chundawat, Associate Professor at the Department of Chemical and Biochemical Engineering, also at Rutgers, the State University of New Jersey. For a complete biography on our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Györgypál and Dr. Chundawat, you may now begin your presentation.
Good afternoon, everyone. My name is Shishir Chundawat, and along with me, I have my former PhD student, Áron Györgypál, who will be talking to you about some of the work that Áron has been involved in my lab and how it relates to bioprocessing. So I'll just give a very brief introduction, and then I'll have Áron take over. So today we'll be talking about the N-GLYcanyzer. This is an automated process analytical system and a platform that's been developed in my lab here at Rutgers to enable continuous manufacturing of glycosylated monoclonal antibodies and biologics in general. So before Áron gets into the specifics, I just want to point out that glycans or carbohydrates are the building blocks of biological systems all around our planet. Obviously, you're familiar with cellulose that covers the planet and other sugars that cover the planet as well. But there are many different kinds of glycans that are found also on the surfaces of cells. And this is just an example of the complexity of glycans that cover the surfaces of cells, but also cover the surfaces of other biomolecules. So what you're seeing over here essentially is an N-linked glycan that's attached to proteins, structurally quite complex and play many different roles. From a biologics point of view, Áron will be talking to you about how we can monitor these to essentially control the glycosylation or N-linked glycosylation in particular, and how that impacts the manufacturing process and some of the tools that we've developed for monitoring N-linked glycosylation of proteins. So with that, Áron's going to take over now.
Yes, so thank you, Dr. Chundawat, for the introduction. So I will be talking about the N-GLYcanyzer system. But before we talk about that, we need to talk about bioprocessing, biomanufacturing. So what we'll be talking about mostly here will be on monoclonal antibodies. So this is a typical monoclonal antibody, and they're used for as combination with immunotherapies. Typically, you will have a Y-shaped protein that looks like this. So it's made out of 4 peptides, 2 what we call heavy chains, 2 what we call light chains. So the purple here is the heavy chain, and then the blue right here is the light chain. So we split it up and basically name them by different parts of the antibody. So we have what we call here the FC region, as well as the FAB region, the Fab region here. The antibody works with 2 different sides. So you have your antigen binding site here, which is typically what connects with a tumor cell or whatever indication that you're looking at, as well as the effector immune response part right here, which will usually complement to a natural killer cell. So to understand glycosylation, we must first talk about what are post-translational modifications. And a post-translational modification is basically what it sounds like. It's the addition of or modifications to the protein after we've already went through the central dogma of DNA to RNA to protein. And glycosylation is the addition of a sugar moiety, so oligosaccharide moiety, long chains, oligosaccharide onto the peptide backbone. So it's a carbohydrate that's attached to the amino acid chain of the protein. And typically it's made out of different sugars. Usually for mammalian glycosylation, we have a typical, what we call a chitose bios core, which is these two N-acetylglycosamines and three mannoses, as well as then decorating up towards different sugars that can be there. So something like this, which is man5, G2, and this could be G2 S2F, for example. So depending on what sugar is there is going to influence things. And the reason why we care about what glycans are there on the antibody and how it's used as an immunotherapy, we need to understand, why this is important. So we can have something that looks like this. This is what we call a high mannose glycoform off the antibody, and typically this is not the best type of glycan to be produced on the antibody, since it usually does not complement well to the natural killer cell, as it will degrade in the bloodstream, because we have lectins in our body that will recognize for those high mannose glycoforms. Then we have something that looks like this. This is what we call galactosylated. So we have galactose off the antibody, and this is typical with high complementation off the natural killer cell. So you have high complementation, high CDC, complementary dependent cytotoxicity. And then you may have something that is fucosylated, so addition of this fucose off the innermost N-acetylglucosamine. And typically this is can cause steric hindrance with the natural killer cells, FCR, so the FC receptor off the natural killer cell. And this usually can cause issues with complementation to the antibody. So usually within companies and pharmaceuticals, we look to produce antibodies which are afucosylated, missing that fucose, but that is a different story for another day. To understand why the glycosylation that we have is important, we must first talk about the glycosylation pathway. So after the protein is produced, it goes into the endoplasmic reticulum, and within the endoplasmic reticulum, you'll have the addition of this sugar moiety. So off the oligosaccharide transferase, which then is what adds this long chain glycan from the dolichol onto the antibody. And from there, you'll have different trimming steps with glycosidases, removing these glucose, removing some of these mannose, and then shifting it from the ER into the Golgi. And typically within the Golgi, we'll start with something that we call man8, which is then trimmed down to man5, and then we'll trim off more mannose and add N-acetylglucose onto it. And from here, this G0 glycoform can become fucosylated, addition of the fucose. It can then add more N-acetylglucosamines, which will then increase the branching, as well as addition of galactose and sialic acids onto it, depending on what is available within the cell at each time. So what can be understood here is that at any point this reaction can stop and kind of be secreted from the with the antibody. So you'll have antibodies that have man8, man5 with just G0, just G0F. And as we talked about prior, this is important because it's going to affect the drug efficacy. Because it affects the ability and the efficacy of the drug that's being produced on this antibody, we typically call it a critical quality attribute. And on the flip side of things, when we're producing these antibodies, we need to be very careful about the culture conditions, meaning the temperature, the pH, the dissolved oxygen, stir rate, as all these things are going to affect the glycosylation as well and affect the reaction that's taking place within the ER and the Golgi. And because of this, we call it a critical process parameter. So any type of parameter within the bioreactor that's going to affect the reaction as well. So what we look to do in our lab is produce what we call process analytical technology. And this is the marriage between the critical quality attributes and the critical process parameters. We're basically looking to understand how changes in the process parameters might influence the critical quality attributes. And the way I like to explain PAT is by some work that was done by Hong et al. In Biopharm Process back in 2020, where he gave this pyramid scheme and kind of explained the sophistication that comes with PAT. So PAT is a mechanism to design, analyze, and control a bioprocess. So at the very bottom of this pyramid, we have trial and error experimentation, univariate analysis, kind of understanding how one variable has an influence on that quality that we're looking at, in this case, glycosylation. From there, we have a little bit more sophistication, which is empirical understanding. And this is basically using statistical tools, using multiple different variables to forecast the outcome. So we might be taking information from past reactor runs and understanding how that how those conditions might be influential on what we're running right now, and seeing how the glycosylation might change with that. Moving up, we have what we call mechanistic understanding, and this is a physical and biochemical principles that do constitute the model-based equations based off experimental data. So this is still a black box approach, but with a little bit more understanding of some of the reactions that are taking place. And to move to the top of this hierarchy, we have what we call first principles. And this is a rather complete mechanistic understanding where we're putting together the reaction stoichiometries, the kinetics, and then and the relationships between the heat, mass, momentum conservation equations. And this is what we're moving towards, but because of this being a bioprocess in which you have multiple different reactions taking place in a biological organism, it becomes almost impossible for us to be able to build a mechanistic or first principles understanding to the level where we're able to understand glycosylation based off of small perturbations in that sense. So as you move up this hierarchy, you have an increasing understanding of the process knowledge, right? So if we have multiple equations that explain the reactions, we can better understand the process. And as we go down, we have an increasing regulatory need of oversight, right? So if we do not understand the process very well, the FDA will be more concerned with what's actually taking place and how our reactions is taking place and how the product is being produced. So this background kind of brings us into the work that we've done here at Rutgers, and most of the work I'll be talking about is from this initial paper from 2021 that we published in Analytical Chemistry, where we were able to use sequential injection to monitor for glycosylation. So sequential injection is a concept where you're using syringe pumps and valving and moving fluids around in the microliter ranges. And this way you can build reactions and you can move different chemicals as needed to be able to automate the process rather than doing it by hand. And typically how we're doing this for antibodies is we'll take a sample from the culture. So you have your antibody and it has glycosylation right here, and then you have other stuff in the media that we need to get rid of before we can actually start the assay. So we'll purify it. So this is typically done with protein A. Then we'll denature it with some types of surfactants and deglycosylate it using N-GLYcanyzer, which is the peptide or the protein that will cleave off the glycan from the antibody. We'll then use reductive amination to add a fluorophore to it. Since the glycans are oligosaccharides, you cannot see them with fluorescence very well, so you have to add a tag to it to increase its sensitivity. We'll enrich it to remove the excess label, and then we'll analyze it again, this time with HPLC. So we'll talk a little bit about the method optimization here. So we identify a glycan sample prep methodology, and the next step is then to actually automate the system versus the offline traditional chemistry. So first thing we did was antibody purification. So we used protein A purification. So you can see here what our protein A column looked like. It's very small. It's about 30 millimeters by 10 millimeters in width, and the resin is only packed to about a diameter of 2 millimeters. So it's a very small column. Here we're showing it in plexiglass, but we found that plexiglass does not work very well. And we moved over to using PEEK, as this is it's a better material to carry the mobile phases that we're working with for our system. And you can see here how well our system is behaving just off of a standard curve that we produce with different bindings. So that now that we've been able to purify our protein, the next step is to denature and deglycosylate. So what we use for our denaturation is sodium dodecyl sulfate, or SDS, which is an anionic surfactant with heat, and that will disrupt the protein, produce the peptides. And then we'll add MP40, a non-ionic surfactant, which will then help stabilize everything so that the N-GLYcanyzer does not get inhibited by the SDS. We'll then add the N-GLYcanyzer and let it incubate, and that will help cleave off the glycans off the peptide itself. So now that we have the glycans, the next step will be to label them. So to label our glycans, we used a traditional shift-based reductive amination using 2-aminobenzenamide, but we also looked to use 2AA, which is 2-aminoanthranilic acid. And you can see here on the right the reaction chemistry that takes place. You have your intermodal N-glycosylation and the rest of the glycans here. And then in the presence of heat, you can add your fluorophore, and they'll produce a imine intermediate. This imine intermediate can at any time be broken in the presence of water to produce to basically produce a 2AB and the free reducing sugar right here. So we have to use a reductant, and typically what's used is sodium cyanoborohydride, which is extremely toxic. So what we look to do is use 2-picoline borane, which is a much more safe alternative to sodium cyanoborohydride. And this is something that's been tested before in literature, but it's something that we've never seen being done under non-aqueous conditions, as this reaction is usually done under, under aqueous conditions, as this chemistry is typically done under non-aqueous conditions. So you can see here some of the experimental work that we've done, and what we've been interested to see is that actually under this aqueous condition, and that's the only way we can make this automated system work is under these aqueous conditions, that we're able to increase the sensitivity of the assay of this complementation or the production of these labeled glycans, and that was basically by using 2AB in conjunction with 2-picoline borane rather than the sodium cyanoborohydride. So next step is to remove that excess label. So you have your sample here with the extra fluorophore. So what we look to use is porous graphitic carbon, PGC, which is a reverse phase material. And in this column, you'll basically have a resonance time distribution of all your glycans. And then earlier on in the chromatography, you'll have the removal of excess peptide as well as the excess fluorophore from the sample. And then you can actually pool all your glycans together. And you can see here some of the recovery steps we have by using a ladder of an oligosaccharide here instead of actually sample, since earlier on we didn't have enough glycan samples to run this experiment, but then we were able to validate it later on. But after we were able to use this PGC enrichment and optimize for it, the next step was to do chromatography. So once the sample was pooled with all our glycans, we're able to run HPLC analysis with that sample by injecting it onto the HPLC. And again, we did this online and offline, and the only difference was that the online methodology used the N-GLYcanyzer system, that's the sequential injection system, versus the offline, which is what I did by hand. What you can see here is that between the online and offline methodologies, between all the glycans that were measured from this antibody, and this antibody, again, this is Herceptin, so this is a biosimilar for Trastuzumab. And you can see here that there's no big changes within the retention time on column, as well as the relative abundances, meaning that we are very robust in the way that we're doing our online and offline methodologies. Now, this brings us to the actual N-GLYcanyzer PAT system and what it actually looks like. So we already talked about the N-GLYcanyzer workflow, right? So we have self-resampling, proteinate chromatography, denaturing the antibody with surfactant and heat, deglycosylating in the presence of heat at 37 degrees, as well as in the presence of P and chase F, the protein that removes the glycan off the antibody, labeling it, and in this case, we were using a 2AB with 2-picoline borane, and then enriching it with a PGC column before injecting and analyzing it using Hillick chromatography. So you can see here the sample will come in from the cell culture with a 0.2 micron filter fiber, and that will go into what we call the Pro Sampler. So this is a system by Fia Labs. This is something that they brought onto market last year. And basically what we're doing here is we're cleaning the sample off the impurities and having pure antibody come off of it. And we're able to actually correlate and build and normalize a feedback control based off concentration, so that we know every single time the concentration of the antibody before we actually start the actual labeling, deglycosylation, and sample enrichment. So we always bring it to about 0.1 to 0.2 gram per liter before we send it to the N-GlyPrep system. And the N-GlyPrep system is where the rest of the chemistry is being done. So this is where you're deglycosylating or denaturing, deglycosylating, labeling, and then enriching prior to injection off the external valve here on the HPLC. And again, this is a completely automated system that's being done automated and autonomously. And you can see here on the bottom what the two flow paths look like for the Pro Sampler flow path and the N-GlyPrep flow path. So the proteinate column, the UV, and we build our feedback control based off this UV from the protein A elution. All the chemicals that we're using, including the calibrant sampling from the bioreactor, which then goes, and then this sample then goes to the second syringe pump here. And that sample is where the rest of the chemistry is going on, like I said. So you have a PNGSF, NP40, your labeling mixture, and SDS all going in to sample and then doing the glycan chemistry, going to this PGC column, and then being injected onto HPLC afterwards. So to talk a little bit about how the system reacts and behaves, we have some data here for that biosimilar of Trastuzumab. So as I said, we were able to measure the titer from the system as well. So you can see here between the online and offline methodologies, there's really no difference. We're reaching about 1.4 gram per liter, 1.3, 1.4 gram per liter. And then all the glycans that were measured off the system, and this is all relative abundances, where you can see not much difference between the online and offline methodologies by maybe a few percent at the larger values and even less when we're looking at the less abundant glycan forms as well. So this is interesting. This is great. Every day we were able to measure the glycan profile. So the next question here that we had was, well, is this enough for us to actually produce process control? And the answer is no. We actually need to be able to measure it more often than daily, at least 2 to 3 times a day, since of course the glycosylation reaction does not change that much every single day, and the amount we're producing is not too much every single day, especially within the first few days. So we need to actually speed up things so that we can actually build process control based off perturbations that we'll do or feedings that we'll do later on as well. So this brings us to the second work that I want to talk about, and this was actually the integration of instant procainamide into the system. And this is a kit chemistry by Adjuvant that I'll talk about in a second, but the way it basically works is this traditional chemistry that we just talked about, which took about 4 hours, 4 to 5 hours, from sampling to purifying, which takes about 5 minutes, to denaturing, which takes 2 hours, to reducing with reductive amination and enriching, which also takes about 2 hours, and analysis, which takes about 1 hour, all being about 4 to 5 hours. With this work that we've done with Agilent and with their instant procainamide chemistry, we're able to bring this from about 4 to 5 hours down to about 30 minutes, which greatly increases our throughput and allows us to have better process understanding. And we'll talk about why that's important in a bit. But basically what we're doing here is purifying the same way with the protein A, but we're denaturing, glycosylating, using a different surfactant as well as a higher concentration of PNGSF, and then at the same time labeling it with within a fluorophore, in this case instant procainamide, which takes about 10 minutes total, and then we can analyze within 20 minutes with our optimized HPLC chromatography. And again, this is just what the chemistry looks like. It's a little bit different than 2AB. It has a tertiary amine, which makes it able to also be used for mass spectroscopy, not just with HPLC. So this is what the workflow looks like with this new integrated IPC workflow, what we call the N-GLYcanyzer 2.0, or at least that's what I like to call it. So your sample is coming off again from the bioreactor with this filtration probe going onto protein A. We're determining the titer with that UV. And then we're left with antibody, about 200 micrograms and 200 microliters, again, about 0.1 gram per liter. We're neutralizing it from that harsh, low pH down back to about pH 8 in a heap solution. So when we bring that up, it brings it to about 50 millimolar heaps. From that sample, we take only a portion of it. We add N-GLYX, which is their Agilent's proprietary surfactant, which will then denature the sample in about 3 minutes at 90 degrees. From there, we'll cool it down to about 50 degrees. We'll add our N-glycanase, or in this case, PNGSF working solution, for about 5 minutes of incubation at 50 degrees C. That'll
Thank you, Dr. Györgypál and Dr. Chundawat, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. Questions submitted today and during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.